All right, so yeah, thanks, Larry. As Larry mentioned prior, my name's Tom Waters. I'm a environmental engineer with US EPA, technical support branch out in Cincinnati. So I'm going to talk a little bit about monitoring for HABs in drinking water sources. I say drinking water sources because we're focusing here on drinking the source water, not necessarily recreational harmful algal blooms, but um, just a quick overview of what I want to cover. I thought I'd organize this presentation into kind of four different categories. Thinking through as if you know nothing about HABs or cyanotoxins, how you can kind of start developing a monitoring program. So one of the first questions that came to mind is what do I analyze? Do I test for cyanotoxins directly? Are there indicator parameters? So we'll talk about that. How to analyze those parameters. So the different methods that are out there, available commercially. How to develop a sampling plan so that, once you kind of identify what you want to analyze and how you're going to do it, how can you, how frequently should you be analyzing? How often, where, and then how to use that data to make treatment decisions. So just to start, I thought it would be helpful to kind of summarize the regulatory environment right now. As everyone knows, these are unregulated on the federal level, cyanotoxins, but there are health advisories out there that were published in 2015. So I'll get to that bullet in a second. But here in Idaho, public water systems aren't required to monitor for cyanotoxins unless of course, you're part of UCMR, which is the federal rule. But of course, you can monitor voluntarily. So I pulled that information from Idaho DEQ's website. I'm sure some of the Idaho folks might be able to clarify if that's incorrect. But and just to give folks an idea of what's going on in some other states in the Northwest US, in Oregon specifically, public water systems that are deemed susceptible to harmful algal blooms, they have to routinely test for total microcystins and cylindrospermopsin. And as I mentioned, there's some health advisories out there. I've got the different levels presented here. In practice, it's kind of a fuzzy message to have two different levels. So I think a lot of folks just kind of look at those lower, those vulnerable levels, 0.3 and 0.7. As the action level, and then of course cyanotoxins were part of the UCMR, the unregulated contaminant monitoring rule. So some systems have been monitoring for those, and there were 10 cyanotoxins as part of that data collection, as part of that effort. So I have those listed here. So just to go into a little bit of detail about Idaho specifically, as I mentioned, there are no drinking water action levels, but Idaho does have some recreational levels. And so there's, if you can see surface scum with any of those cyanobacteria that are known to produce toxins, there action needs to be taken. And then there's some cell concentrations where different actions need to be taken. So those are just, and I'm just providing these to give folks an idea of what states and on the federal level what needs to be monitored for, and kind of what the ballpark figures are for taking action. So moving on to Oregon, they're a little bit more prescriptive about what needs to happen with drinking water monitoring. And so basically, just to kind of summarize their monitoring regulations, I won't go into a lot of detail here, but they have to, systems in Oregon have to monitor seasonally in their raw water, and depending on what they find, if they do find cyanotoxins above that 0.3 level, or if there is a recreational advisory, they need to do some entry point, so some finished water sampling to confirm that it's not passing through treatment. And so I've got some actions listed here under entry point sampling if it is detected. but usually two consecutive samples, depending on your frequency, get you back to the regular monitoring. If you have found it, and then you start, optimizing, and then you're getting non-detects, two consecutive samples will get you back to just raw water sampling. So moving on now. So that's just kind of what's in the region, in the Northwest. I know Ohio has some monitoring regulations as well, but really it's kind of it's an unregulated contaminant on the federal level. So it's kind of up to the individual states and water systems, on a voluntary basis to look for these and to think about how to treat them. So as far as the way this presentation is structured, as you recall from the beginning, I was going to start talking about initially what to monitor. And so I've got listed here some source water and weather conditions that, aside from any kind of analytical or taking samples, these conditions can suggest a higher chance of HABs forming in a water source. So those can include, excessive nutrients. If your water body is located next to a lot of agriculture that has runoff, that could be a risk factor. Hydraulics in your water body, obviously calmer, stagnant waters lend themselves to being more conducive to cyanobacteria development. And then in terms of weather, warmer water temperatures, particularly above 25 degrees C, can kind of select for cyanobacteria as opposed to some of the other phytoplankton that are in the water bodies. Light intensity, so obviously, long periods of sunlight, because cyanobacteria are photosynthetic. They make their food from sunlight, like plants. And then, rainfall can wash nutrients and other food for the cyanobacteria into the water. So if you have, periods of rainfall and then long periods of sunlight, that can be a risk factor for cyanobacteria. And then wind. Wind can kind of concentrate some of the scums into certain areas of lakes, depending on the wind patterns. And, especially if there's an intake on a certain portion of the lake that's, on the downstream end of where the wind's coming from, that can be problematic. So I have organized a summary. I kind of try to think of all the different ways you can monitor for cyanobacteria, and I've got them kind of listed in different categories here. So I kind of try to make an attempt at making these, starting with visual observation. That's probably the easiest, least expensive way to monitor, and then moving on to probably some of those analytical methods where they're more complex and costly. So I'll talk about each of these boxes in separate slides. So this is just kind of an overview. So visual observation, then we'll talk about some indicators. So, not measuring cyanobacteria or cyanotoxins directly, but what are some commonly collected water quality parameters that could suggest maybe needing to actually sample for the cyanotoxins. So pH and temperature can be indicators, taste and odor. Even if you're having decreased filter runtimes in your treatment plant, increased chemical usage, and then kind of moving on past the indicators, some states and water systems have had success actually identifying cyanobacteria cells and counting them. As you saw earlier, recreational levels are based on cell biomass or cell concentration. So in that situation, it would be helpful to know, what types of cyanobacteria are out there, how much of them are out there, because different types of cyanobacteria produce toxins and some don't. So it's helpful to know that. And then finally, analytical methods. There's a couple of methods out there for actually measuring the cyanotoxins, which are that's what we're ultimately concerned about, from a public health perspective. And then down on the bottom here, I've got a couple other approaches to monitoring. Satellite imagery has become kind of a promising tool, especially on larger water bodies. There's some indicators of cyanobacteria cell, aside from actually looking at it under a microscope. You can kind of deduce how much biomass is in the water based on turbidity, pigments that are produced by the cyanobacteria, organics, and color. And then finally, I'll talk a little bit about the molecular methods, qPCR, polymerase chain reaction. One state in the U.S. it actually uses that, and so I'll talk a little bit about that too. So starting out with the visual observation, this is I took these pictures and these ideas from a couple states have talked about these. Just real simple tests, a bottle test and a stick test. I've got the states listed down here. There may be more, but these states mention them directly on their website. So a bottle test, basically just taking a clear bottle and allowing it to sit for a while. And you can kind of see the progression here in the pictures. You know, you get your sample of water. It's got all your different types of, you know, it could be cyanobacteria, it could be algae, it could be diatoms, it could be you know, a mixture of them. But if you leave it sitting for a while, you can see here that they've kind of all moved to the top, and that's because cyanobacteria, they have the ability to regulate their buoyancy. They can go up and down in the water column to find the optimal levels of sunlight so that they can, you know, they can make their food. So the thought here is that if you're unsure of what you have in your water, this test could kind of tell you, you know, regular algae would just kind of hang out, looking like the first picture. But if they do, after time, move to the top, that could indicate that this is cyanobacteria. And then also a stick test. This is a picture I grabbed from, I think it was Colorado's website, just dipping a stick in the water and pulling out some of the green matter. Usually cyanobacteria don't look like this. There is one kind that does, but for the most part, the stick should just pass through if it is cyanobacteria. But if you're pulling up large filamentous kind of algae like this, it's probably not cyanobacteria. The odds are it's probably some kind of green algae that's not going to produce toxins. So those are just kind of some high level screening kind of tools you can do with, you know, minimal equipment, minimal cost. So I'll talk a little bit now about satellite imagery, and this is kind of you know, in the last maybe 5-10 years, it's kind of emerged as a good tool as, you know, more satellites go up and there's, you know, better software and tools to process the aerial imagery. So I've got I pulled this. I think Larry showed this figure as well. Just kind of the status of the different water systems out in Idaho, and I got the website listed down here. But and on the right is I just pulled some pictures from some of the satellite projects that are going on. These are a combination of the two projects. And I'll talk more about these, but this is kind of what the satellite imagery will look like. You can kind of see the, you know, as you get the red colors, that's higher concentrations of cyanobacteria. And I'll talk a little bit about how they process these images to determine that. This top one is closer to where I'm at here in Ohio. This is Lake Erie. This western basin of Lake Erie is kind of notorious for cyanobacteria, and this was the area where a bloom occurred that caused the Toledo incident, if you're familiar with that. So basically, as I said, there's two projects that I'm aware of. There could be some others out there, but two projects that are working to analyze satellite imagery and use that imaging to you know, determine real-time cyanobacteria concentrations and also aid in forecasting when you combine that with wind forecasts and currents in larger water bodies. So NOAA has a program just on some selected lakes across the U.S., bigger lakes, you know, that have you know, several water systems pulling from them: Western Basin of Lake Erie, Green Bay, Chesapeake Bay, et cetera. There's several here. And then another project that I'm a little bit more familiar with, just because I work for EPA, is the CYAN project. It stands for Cyanobacteria Assessment Network. I've got there is a smartphone app, which is pretty helpful for going out in the field and looking at, you know, you can click on individual pixels in a satellite image and it'll estimate the concentration of cyanobacteria. I've got that hyperlinked here so that you can check that out and maybe download it if it's helpful for anyone. But basically what this project does is it uses chlorophyll A and phycocyanin to correlate to cyanobacteria biomass, and software can analyze, you know, emission, light emission spectra that can be correlated back to a chlorophyll or phycocyanin concentration, and then that can be correlated to a cyanobacteria biomass. The big limitation of this this tool is that it's really applicable to larger water bodies that are the current resolution for this is a 300 meter pixel. So that comes out to be about a 22 acre minimum size water body to resolve that water body and be able to get good estimates on cyanobacteria biomass. So just talking a little bit more about that Cyan project. These are all, I've got a map here from the website. These are all the quote unquote resolvable lakes, the ones that are large enough to see on the satellite and estimate cyanobacteria concentrations. So you can kind of drill into this more. I've got a paper cited here that talks about this, but what they've done over the years as they've developed this software is try to correlate, you know, what they're finding, what they're estimating with the satellite with actual, ground truth samples. So the in situ cyanobacteria abundance. So I've got this figure here from the paper, and they do have a pretty good correlation, you know, an R squared of 0.95, you know, correlating the satellite cells that they're estimating with the actual in situ cyanobacteria. So they've done a lot of ground truthing for this. It's a pretty good tool, but again, the limitation is water body size. So I'll move on now. We've talked about satellite imagery. We've talked about some simple tools that can be used to determine if you have cyanobacteria. So now I'll get into some of the indicators. As I mentioned, you know, you can think about, you know, turbidity as a tool that might indicate some increasing cyanobacteria, especially if you have a pretty stable water body that you know, raw water turbidity. So and same with pH. As we've kind of mentioned, you know, cyanobacteria behave like plants. They take sunlight and convert that to sugar as their food. So while they're doing that, it's, you know, it's called respiration. And while they're doing that, that can the flux of carbon dioxide that they're taking in and they're giving off, it can affect the pH of the water. And so that can also be an indicator. You know, if you see a swing in pH, an increase in pH, that can be an indicator of, you know, cyanobacteria starting to produce. So here I got two plots talking about turbidity and pH. This was from I took this data from a plant. It was in El Salvador that I worked with. They had a huge issue. They pulled a HAB into their treatment plant, and they had no idea, and it passed through the treatment plant, and then they got all these complaints about taste and odor and stuff, and they asked for help. And so, you know, I went down there and I was looking at their data, and this was, a lot of you can probably guess when they started seeing issues in the plant and out in the distribution system, because, you know, they, for the most part, they're between 10 and 15 NTU. And then all of a sudden, you know, this was in January of 2020. I think it was on the 14th of January that they pulled this bloom into their treatment plant. And, you know, you can see the turbidity spike here, indicating that, you know, that was probably a lot of cyanobacteria that they pulled in. And same with pH. You know, they have a swing of almost a unit in their raw water pH that coincided pretty closely with the incident. And so I thought that was kind of telling, you know, a good example of just using simple indicators to kind of back, you know, retrospectively analyze, you know, what happened there. So I talked about a little bit about indicators, and now I'll talk a little bit about, you know, I'm not a phycologist or a biologist or anything, but, you know, some systems, at least in Ohio, I know that they've had their operators go through, you know, some training on how to identify cyanobacteria cells, how to count them. So this is a picture of some Microcystis cluster of cells, and I think there's some Anabena or something around here. But yeah, this was at a plant. I grabbed a screenshot of and the operators were able to identify it. So knowing you know what's in your water specifically about cyanobacteria can kind of help you understand what toxins might be produced. You know, if there's quite a bit of them, you can probably deduce that cyanotoxin concentrations might be higher. So it's just a helpful tool to help kind of help direct your treatment adjustments if there is a HAB. And I will talk a little bit about, this kind of falls into the category of direct measurement, but gene detection through PCR. State of Ohio's had some success with that, so I'll talk a little bit about that next. Oh, what? No, I've got another slide on cell identification. So this was going back to that El Salvador example. They did a kind of a post-event analysis, and this is what came back with all the different genera of cyanobacteria. So you can see Planktothrix, Microcystis, Pseudobena. So these are all known toxin producers. The concentrations are a lot lower than the classic definition of a bloom. I think Idaho's recreational bloom sort of action levels 40,000. There was something you had to do, and then 100,000 cells per mil. So these are lower. I mean, this was post-event, so it's not the height of the event, but you can see that there are toxin-producing cyanobacteria in the source water, and so that helps again understand what help understand what cyanotoxins might be present if these cyanobacteria did bloom and did proliferate. So yeah, I was going to talk a little bit about PCR. Ohio has had a lot of success with this for specifically as a predictive tool. As part of their monitoring regulations, they ask systems to analyze qPCR samples too. So it's a good screening tool. It has, from a time perspective, it can kind of, you can see these genes prior to any spikes in cyanotoxins. So I pulled these figures. They're pretty complex, but I'll try to make them simpler by just talking about colors here. So the blue and the red are qPCR. So that's quantitative polymerase chain reaction. So the name, don't worry about the name, but basically what it does is it identifies and quantifies the presence of total cyanobacteria, along with it can quantify the genes that specifically the genes that are in the cyanobacteria cells that are responsible for the production of cyanotoxins. And so that's important because you see these in all of these are different sampling locations in an Ohio water body, and you can see that they observe spikes in the qPCR genes, the toxin producing genes, and that's shown in the blue and the red, different methods. Much earlier, it's almost a week prior to you seeing a spike in actual microcystin concentrations. So that's the black and the green lines here. So each plot at each sample location, you can see they had spikes in microcystins, around mid-July in this plot, but they saw spikes in qPCR, almost weeks earlier. So it's a good. Ohio has said it's a good predictive tool. Systems can kind of take action, knowing that seeing that the increasing toxin producing genes, they can kind of get a feel like this bloom's up and coming. We probably need to take some treatment steps. But I'm really simplifying this study, but if anyone wants to get into more of the details, I've got the paper cited down here. So now we've talked a lot about indicators. We talked kind of like dancing around the cyanotoxins, which is the main public health concern. So there are methods that are out there. You can analyze directly for cyanotoxins. But before we get into those methods, I thought I would just take a minute to kind of make some definitions. And Larry mentioned these earlier. Intracellular were basically meaning toxins that are produced by the cell, but they stay inside the cell wall. Extracellular toxins are those that have been released from the cell. They're dissolved. They're dissolved in the water outside the cell. And then combined is just the sum of both of those. And basically, we'll go through some of the methods. But when I say intracellular or extracellular, it really has nothing to do with the methods themselves. It's how you prepare the samples. And so the way you do that is the methods are identical, but you take two parts of the sample, two aliquots. One you put through a freeze-thaw cycle, which causes the cells to lyse or break apart and release the toxins. Then you filter that to get all that cellular matter out, and then you perform your method. And so that gives you a combined reading for cyanotoxins. It's both, everything that got burst out of the cell plus what was in the water to begin with. And then the other part of that sample is just filtered. So you just take the cells out prior to lysing them, and then you perform your method. And that gives you the extracellular concentration. And then just by simple subtraction, you can figure out what was contained within the cell. So the combined minus extracellular is intracellular. And this has a lot of relevance to how you treat. Larry's going to talk a little bit about that later, but think about a cell as like a particle, but extracellular toxins are more dissolved. It's molecules, and so particle removal technology is conducive to removing the cells, while other technologies are more conducive to taking out the molecules. So I've got these slides are they're going to be busy, but I'll try to just make the main points. Just comparing the different methods that are out there for analyzing for microcystins. That's kind of the most commonly found. It's the most researched cyanotoxin that's out there. So there's several different methods, and I'm just including some of the kind of the main points of the methods here. So ELISA, and I'll talk about these in more detail, but just to give an overview of what to expect. The main difference, so ELISA is an assay, and they're available, in strips. You can get them in tube strips. Their limit of quantitation is pretty high relative to like the health advisories, but you get a quick result. And there's a lot of interpretation with those. It's basically just kind of like a pool chlorine tab. You're comparing colors. Whereas the same assay chemistry, there's also like a lab method that's much lower limit of quantitations. You can get lower concentrations on that, but the time takes a little bit longer. And there is an EPA method 546, which was written for UCMR, so that's out there and available. And then finally, liquid chromatography, mass spectrometry, an order of magnitude lower quantitation limits. It's a lot more selective. The timing takes, if you have the equipment, it takes less than a day. But if you're setting the sample out, it could be weeks. And this is a method for that. One of the other limitations and the differences between the liquid chromatography and the ELISA assay are, the assay gives you an estimate of total microcystins, whereas the liquid chromatography, I said it, even though it's more selective, it can only quantify what you have standards for. And so EPA method 544 is only 6 specific microcystin congeners. So if you've heard like microcystin LR, RR, YR, there's all these different congeners for microcystins. But when you're doing liquid chromatography, there's only for this method, there's six standards. I think there's another modified method that's got 12. So if you're looking at your microcystins concentrations and you're trying to make decisions based on the health advisory, that's written as total microcystins. And so this, it's just to keep this in mind. If you're trying to compare them, they probably won't compare directly. And obviously I'll talk a little bit more. There's other advantages and disadvantages of using each method. If you're concerned about cylindrospermopsin or anatoxin, there are assays for that. Kind of the same advantages and disadvantages. The timing, the limits of quantitation, it's pretty similar. What makes ELISA different here is that cylindrospermopsin and anatoxin don't have all of the hundreds of congeners that microcystins do. So these are a little bit, these two methods for these cyanotoxins are a little bit more comparable because you're not missing congeners if you run LCM SMS. So liquid chromatography, just listing the methods here that have been written by EPA. There's 2 for finished water. So it's important to pay attention to the applicability of the method if you're sending these off, or if you're fortunate enough to have these expensive equipment in your labs. EPA method 544 is for 6 microcystins and nodularin. EPA method 545 is for cylindrospermopsin and anatoxin, and that's for finished water. There are some ambient water methods, so that's like your raw water, and I have them listed here. These are a little bit different in that they reference EPA documents. This is the ambient water method that's got 13 selected microcystins, and so a little bit more than the finished water. And then ELISA, I mentioned it's an assay. That long term stands for enzyme-linked immunosorbent assay. Separate assays are used for different groups of cyanotoxins. So there's different assays for microcystins. There's one, available for cylindrospermopsin, saxitoxin, anatoxin. But again, they quantify total microcystins. So, and they do that by detecting like a portion of the microcystin molecule that's common to all the different congeners. And another one thing to keep in mind too, is that you think about the ranges of this method. The calibration curve isn't linear. It's sigmoidal. It kind of looks like an S, a flattened out S. And so at the low and higher ranges of the method range, you're not going to get as much resolution as you would if you're on the linear range. So just something to keep in mind, especially if you're like a lab person or you're interpreting lab results. Just some caveats to kind of keep in mind. A little bit more detail on comparing the methods. So as I said, EPA method 546, that's the assay. That'll give you total microcystins. EPA method 544 or any other the other liquid chromatography methods, they're much more sensitive. They're gonna, you can detect lower concentrations, but it's for specific microcystins and not the total. And so I tried to list some of the limitations out here for that. So, I mentioned non-typical calibration. Lab technique is really important for for the assay. Your pipetting technique kind of plays into how accurate your results end up being. And so, yeah, I think I've kind of covered this already. Early on when cyanotoxins were becoming a bigger and bigger concern for water systems, we kept getting questions about, I've split samples, I've sent some for ELISA analysis, I've sent some for liquid chromatography, and they're not matching up. Like, which one do I make my decisions based on? And it's a hard question to answer, but I think just for folks to understand the differences between these methods, it's helpful to interpret those results. And so, a lot of times liquid chromatography, you only have, if you're running that EPA method 544, it's only detecting 6 of the microcystin congeners. And 546 is going to tell you, the assay is going to tell you the total microcystins, with varying, if you're trying to get specific, with varying degrees of assay recognition. So the whole thing is calibrated on one congener. So there is a little bit of cross reactivity that can occur. And so, it's just important to, none of the methods are perfect. They all kind of have their place. They have their advantages, but also disadvantages. And so it's important to kind of understand that as you're interpreting results. So this is a busy slide, but I thought it'd be helpful to include. It's actually from we did a workshop in Iowa a couple months ago where we went through all these methods for the workshop attendees. They were state folks from across kind of the Midwest and South. And we went through the methods. We kind of provided this sort of overview, and then they had, a hands-on workshop, and we asked them for feedback at the end about, how applicable are these methods? How could you use them in the field? And this is the feedback we got. I'm just going to go back real quick, but I'm not going to read through every part, but I thought it would be helpful just to kind of summarize what your peers thought of these methods as well. And so just kind of high level. We introduced a SOND. These are commercially available, like YSI has them. And they measure chlorophyll, phycocyan. And so those are the pigments produced by cyanobacteria, along with some other, ancillary parameters here. And so, I've tried to include some costs in these, about $15,000 for that. Good as a real-time monitor, but you're not measuring cyanotoxins directly. Fluorometers, they can measure chlorophyll and phycocyan, and the 2 pigments that are relevant to cyanobacteria. Again, it's, you can kind of correlate it to cyanobacteria biomass. It's a good screening tool. about $5,000 up front for a meter like that. But again, you're not measuring this, cyanotoxins specifically, but these are good screening parameters. I think we discussed probably enough about identifying cells, but there are some training requirements. I don't know what I'm looking at if I look at a picture of cyanobacteria. So you need a biologist or a phycologist, and you need a microscope, specialized equipment. But it is good for understanding which cyanotoxins to ultimately test for. And so we're keep moving on these methods, and again, this is just based on feedback from peers. So I thought it'd be helpful to include. There is an automated system to do the ELISA assay. Super expensive, but it kind of takes all the human error out of running the assay manually, which is down here. So it's, if you're doing it manually, it's cheaper than buying the automated system. But obviously more expensive to get the automated system than just run it manually. Finally, test strips. We kind of look at these as screening tools. They run the same chemistry as the assay, but their limit of quantitations higher. And I think the way the industry kind of looks at the test strips is they're more used for screening. So if your test strip comes up positive, basically you probably should send some samples off for more detailed analysis like liquid chromatography or ELISA. And then finally, there's a new, and I don't know too much about this, but there is a Hawk makes what's called a light deck, and it's intended to be a quicker, I've got 10 minutes listed here for single samples. It has if you're familiar with the SL1000, it has keys that are kind of similar to that are the consumables. And it automatically does your quality control. Pretty kind of a high upfront cost, and it ends up being about $27 per test. And so the feedback from the group was that it's kind of promising. It's quicker than running ELISA, a little bit expensive, but it's new, and we don't have a lot of experience with it. But I just thought I'd mention that. Our group purchased one, but we haven't used it yet. Hoping to get some more information. And be able to relay that to folks like you all. Okay, so I kind of went through the gamut of or the spectrum of all monitoring methods that are kind of common out there that are commercially available. But now that we've kind of talked about what to analyze, whether it's an indicator or cell biomass or directly measuring cyanotoxins, we've kind of talked about the advantages and disadvantages of the methods. So what, say you understand that, where do you sample, how frequently, and how you use the data. And so I've included this slide. I think Larry's he's gonna talk a lot more about the treatment end of things next, but I just thought I'd include this because it's not, you're not just limited to taking samples in the raw water and the finished water, like the way UCMR prescribes or the way some of the state monitoring regulations prescribe, but it is helpful to know how each unit process of your treatment plan is performing at removing these cyanotoxins. And so checking my time here real quick, sorry, I think I'm kind of running out of time, but we really emphasize in our optimization trainings that that multi-barrier treatment strategy. And so how do you know how your different barriers are performing at removing cyanobacteria cells and cyanotoxins? You take samples at at each unit process, and so we call that a plant profile. And so how do you develop a plant profile during HAB? It can be done with grab samples, and you analyze the samples with the methods that we've talked about previously here. I did mention a sonde, and maybe I should have included this slide prior to that other table, but there's commercially available sondes that have sensors for chlorophyll A and phycocyanin, as well as pH, temperature, and turbidity, and other parameters. But chlorophyll A, is a you probably heard that in your biology classes in school. It's a pigment that all photosynthetic organisms use to make their sugars, make their food. Phycocyanin is a pigment that is unique to cyanobacteria, and so collecting information on both these pigments helpful because you can kind of differentiate between what's, the total photosynthetic biomass in my water body, and then phycocyanin can tell you what specifically, is cyanobacteria. And so the SONs can they have calibration curves, and they can provide information on that. So you can use a SON to do a plant profile. And the picture here is taken from a project in Ohio that we did, and the guy here on the left is holding the SON down in the filter, one of the filters, taking top of filter parameters that are listed here. And so basically you can just take that SON and go around and dip it in all the different processes, and you can develop a plant profile. You can do the same thing with grab samples too. So it's not, you don't have to have a $15,000 SON to do it. But these are some suggested spots to sample in the plant. Obviously your raw water and your filter effluent are very important, but it is helpful to see how your sedimentation, your clarification processes are working as well. And I'm just going to show up 2 plots here to kind of emphasize the value of doing that. This is the data from that study that I'm showing from the picture here. We used the SON to collect chlorophyll data, phycocyanin data, and then we pulled grab samples and analyzed for microcystins. So you can see kind of the main message here is that this is the kind of plots you can make with doing a plant profile, and you can see that how effective the coagulation, flocculation, sedimentation process is at removing cyanobacteria cells. And so we're using that chlorophyll and phycocyanin as indicators for how much cyanobacteria is out there. And it does kind of correlate to the toxin concentrations as well. And I think the main takeaway here is, you've got your raw water, your chlorophyll is pretty high. You've got high total microcystins, but as you pass through those clarifiers, those numbers really drop off. And so Larry will talk more about, the different treatment processes, but this is, you could conclude from this plant, maybe I should spend a lot of time optimizing my clarification because, I'm getting a lot of removal right there. And the same for this plot. This was from a study on a Lake Erie system using chlorophyll as an indicator for total biomass of cells. Did 3 sampling events, and you can see that most of the cell removal is accomplished prior to filtration. And so it's helpful to know that and help direct your your optimization efforts. So that's it, Larry. I apologize. I think I went a little over time, but
no problem, Tom. For some reason, I'm having a problem starting my video, but can you hear me?
Yes.
Okay. No problem. I was going to give you a little extra time anyway, so we'll extend the break a bit. There were 2 questions. So just a couple comments. There's just a lot of information in Tom's slides. So a lot of that's intended as reference materials to go back and review. You'll also have an opportunity in the smaller groups to ask questions. So if you don't get them all answered here, feel free to bring them up in the smaller group. There was a question from Bonnie. Can ELISA be done in-house, or does it need to be sent out? Can you comment on that, Tom?
Yeah, that's a great question. It can be done either way. It does require a plate reader, if you're familiar with that. Basically like a benchtop piece of equipment that connects to a computer. So you run this assay. I wish I had included some pictures now that I think about it, but you run this assay and then you put it in a plate reader. So it does require a little bit of capital investment for that plate reader. But and the other thing I'll say about doing it in-house is it's pretty, the ELISA technique is pretty user dependent. And just from personal experience learning it and trying to run it, it's very sensitive to how quickly you can pipette the different channels, how good you are at pipetting, and so it takes a lot of practice. I'm not good at it, and I still can't get good quality control on it. So it probably requires some training, and as I mentioned, a little bit of investment for the plate reader to do it in house. It definitely can be done in house. There's systems that do it. But I think for the majority of smaller systems that don't have the lab capacity, the samples are sent out.
Okay, second question, just a kind of a basic question from Adriana. What is an assay?
Yeah, that's a good. So I'll just preface this by saying I'm not a chemist. We do have chemists in our group, and so I can, if there's further questions, I can pass them on. But basically, it's what it is it's a plate that has small kind of sample cells. They're less than a centimeter in diameter. And what you do is part of the plate is dedicated to calibration. So when you get a kit from a manufacturer, it'll have some standards in it, and you put those in those wells. I mean, they're called wells, the little sample cells. And then you put your unknowns in the rest of the cells, and you run them. Each well, each sample gets 2 wells, so you're running them in duplicate. And then there's some wells dedicated to quality control. And so this is all developed on it's like a plate with these small little sample wells. And you go through a series of steps where you add reagents progressively to cause the different chemical reactions to cause the detectable compounds to be present that the plate reader can read. So it's kind of like it's almost like a statistical way of determining the concentrations in the different wells. And it gosh, I wish I had a picture. But yeah, I mean, the principle is like it's called a competitive assay. And so there's I'm blanking on the terminology, but basically there's you're coating the insides of the wells with certain reagents that bind. If there are cyanotoxins present, they bind with them, and then they react with subsequent reagents you put in, and it makes a color. Basically, the color of it's like a blue color, and then you analyze in the plate reader, and it's inversely proportional. So if you're just looking at it, the assay visually, the wells that have the least amount of color have the most cyanotoxins. I don't know if that helps.
I think we have a video too, Tom, that's going to be in the materials on the method.
Yeah,
so I'll just mention that in the download link, there is a video on how to do the method if you're interested in kind of getting more specifics. So
yeah, it's only 7-minute video too, so it's kind of a quick one. It's a great overview of how to conduct it and what different equipment's required.
With that, we'll go ahead and close out the first segment, and we're going to go ahead and take a break. I've extended the break a bit. We'll be back at 10:40 Mountain Time. So go ahead and rush off and take a short break, and we'll be back to start in our next segment before lunch.